My name is Yuan from the Fabrication. And my presentation topic today is constructivities in a microfluidic device. Here is the outline. I will talk about the background, design testing of microfluidic device, the drug model preparing, and experiment results. Also come with the conclusions. In biochemistry and chemistry lab, we normally use micropipette and microtiters to perform experiments. However, normally those experiments need to be carried out several times, like hundreds or even thousands of times. That will be quite time-consuming and a lot of labor work. To address these issues, we introduce multi-channel pipettes or robot systems. But for the multi-channel pipette, output is still not that high, and for robot systems, the setup is a little bit complicated and relatively expensive. Here we have other options. We can also use droplet-based platform, and this table compares disadvantage and advantage of these two platforms. For the traditional platform, which is most widely used platform, it's very easy and simple to use. However, it's relatively low throughput. The consumption of each test, normally it will cost a few microliters range. If the sample is very expensive, that will cause some waste. But for droplet-based platform, it has very high throughput, and the consumption of each test is only a nanometer. Also, it's automated operation system. Our previous work, including generating those droplets, observing their fluorescent image on the microscope, and also control the drug release within each droplet. However, those designs cannot trap those droplets in a certain place and monitor the drug release in real time for a long period of time, which will be critical in the future to observe the drug effect on a single cell. Here is our new design. We integrate more functions into a single chip. It generally has 4 different parts. The first part is the T-junction used to generate droplets. Second part is the merging function used to deliver the control solution. And we have a switching channel to switch on and off the storage chambers. And also we have storage chambers to trap those droplets in order. The fabrication process is quite straightforward. We just clean the glass slides using S-weight photoresist as a model, and we pour PDMS, curing, peeling off, punch through to get the inlet and outlet. And when bounded with glass slides, ship the final result. Here is the fabricated device. It integrates several functions, and I will introduce each one of them. First part is the T-junction. Those T-junctions are used to generate the droplets, and each droplet will be served as a Microtiter work. And those images will show that how one droplet is generated by using the oil to cap the drug sample solution. Now we have the drug droplets, and we need to deliver control solution into each droplet. In those photos, we can see that the control solutions come from the right side of this chamber, and the sample solutions come from the top. When these two kinds of solutions met in the merging chamber, they will merge together and form a bigger droplet. And after that, the control solutions deliver the result. Now, in order to make sure that each storage chamber will be occupied by only one droplet, we have to design a switching chamber. This is because normally it takes a while before you can generate stable droplets without any bubbles. And we don't want these bubbles or ununiform droplets go inside the storage chamber and occupy it like this image shows. And when we design the switching chamber, we can easily filter out those bubbles or ununiform droplets until we can generate uniform droplets. And this one shows how each chamber is occupied by only one droplet. Those photos show how the switching chambers work. By carefully designing the flow resistivity, when we switch on these switching chambers, all the ununiform droplets and bubbles will flow towards the switching channel like this. But when we can generate uniform droplets, we will switch it off. And after that, all the uniform droplets will flow towards the storage chamber like this. On the last part, the storage chamber. As a label, the first chamber has been occupied by the first droplet. And when the second droplet comes, it has 2 options. You can go inside the storage chamber or flow towards the second one. However, the first chamber has been occupied by the first droplet, So this branch has been blocked, leaving the second droplet no choice but carry on flow. And so as the third one. And when the second droplet comes to the second chamber, which is empty, it will go inside and also block this chamber. So when the third droplet comes, it will flow towards the third chamber, and so on. And now we could put the first droplet in the first chamber, second one in second chamber, third one in third chamber. And why do we need trapping those droplets in order? This is because during the control solution delivery step, we could easily control the testing conditions, like verify the amount of control solutions we deliver into each droplet. But to monitor each testing result, we have to identify each droplet and figure out which one is which one. So by trapping those droplets in order, that's not a problem. Those photos show a demonstration experiment about the drug model release in our device. We choose manganese carbonate microparticles as a drug model, and we cover it by layer nanoassembled microcapsules. To control the drug model solution, we use a control solution, diffuse the cores, and dissolve the cores. And that will be fused out of microcapsules and leaving the shell only. Before we flow those drug sample into our device, we do the equivalent experiment on a glass slide. Those images show that, the first one shows that a few seconds after we deliver the control solution into the drug model, the color is still quite dark, which means that the core hasn't been dissolved. The second one shows that after 5 minutes, most of them dissolve, but still feel dark, which means the core hasn't dissolved. And the third image taken after 10 minutes, you can see that all of the microcapsules are transparent. So we flow our drug models into our device. Two images on the top shows that without delivering control solutions, the core will not be dissolved, even we leave it for a long period of time. And those three images on the bottom show that after we deliver the control solution into our droplet, like this droplet, as time goes by, the core will be dissolved gradually, and it will become transparent in the end, which shows that all the cores have been dissolved. Those curves show that we can, by changing the control solution's concentration, we can verify the release rate of this drug model. And as we increase in the concentrations of the control solutions, the dissolve time will be reduced from less than 10 minutes to less than 5 minutes. To sum up, we introduced a new microfluidic device, integrate several functions into a single chip. We also store droplets in order for a long time for monitoring and testing. And we also use manganese carbonate microparticles as a drug model to demonstrate the control release in droplets. Thanks a lot for listening.